Welcome Miriam and Don. They're no strangers to us. They've been running the morphometrics workshop every summer for how many years?
Something like 15.
15 years. It is really well attended by students from across all the museums and associated departments. And Miriam has been something of a force in studying morphometric evolution. And that relates to the topic today, unpredictability of phenotypic evolution, Sciurid responses to climate change. Thank you.
It's an amazing experience to be here and giving a museum lunch. Because the last time I did this, we had slide carousels. And that's how long it's been. And it's not a tag team talk. I kept threatening Don that I was going to decide he was going to give it, but I won't do that to him. Especially since he's the one who drives. So, there are three possible responses to climate change. A population species can go extinct, they can move to track their habitat, or they can adapt either via evolution or plasticity. Now, habitat tracking has been well documented on sort of general surveys and summaries of the literature. That in the northern hemisphere, elevational and latitudinal boundaries for species have shifted. And I figure many of you know this literature as well as I do. Adaptation has also been well documented, especially changes in timing of periodic life cycle events. And there are some just cool examples showing how spring has shifted in its time. There are also indirect effects of climate change. Since whole communities do not move in concert, and whole communities do not shift phenologies in synchrony. So, people have talked about community disassembly, disrupted interactions between competitors and or predators and prey, and mismatched phenologies across trophic levels. Now, the Grinnell Resurvey project has been demonstrating idiosyncratic responses to climate change. So I'm not going to go through all the slide. The point is that not all species shift their ranges by the same amount in synchrony. So the zonational boundaries have been changing over time. So what are the expectations? Species that track their thermal habitat will still confront novel biotic environments. So they're tracking only one dimension of their habitat. They're not truly tracking their habitat. And responses to novel biotic environments will likely be manifest in altered trophic morphology because there's shifting interactions with predators, prey, and competitors, and diet shifts that affect the material properties of food, which if you live in a world that's focused on mammals, dietary consistency is one of the critical determinants of trophic morphology. Particle size, hardness, and toughness especially. So the question is, how predictable are these responses? Can we predict phenotypic responses to climate change? And if so, by what can we predict them? Can we predict how much they will change? And can we predict what will change? Now, we know that trophic morphologies can adapt very rapidly. Darwin's finches are a classic example of exceptionally rapid morphological change. In 1977, the drought conditions favored larger birds with deeper beaks. And in one generation, beaks deepened by 5%, or by the measure that I'll often be using in this talk, by 0.657 standard deviations in a single generation. Such rapid change is costly. The population that evolved so rapidly crashed to 15% its initial size. So rapid evolution on the order that we say in Darwin's finches can drive a population to extinction rather than protect it from extinction. And rapid response to fluctuating environments can be extremely costly. Darwin's finches reverted. So wet conditions favored larger birds with shallower beaks, and in just one generation, the beaks shallowed by 0.372 standard deviations. So what rates can be sustained when the climate and the environment is continually changing and you experience transitory fluctuations in the environment superimposed on top of the sustained change? So Berger and Lynch, who developed a relatively complex but still probably unrealistically simple model. It said maximum sustainable rates for populations in a continually changing environment subject to fluctuations in the optimum. 0.01 standard deviations per generation. Now, plasticity may reduce the risk of extinction. It can allow populations to persist despite fluctuations in the environment, since plasticity may respond to those fluctuations in the environment, enabling the populations to adapt to the sustained environmental change. But plasticity can also retard evolution, because if selection is favoring the morphology produced by plasticity, there won't be any evolutionary response to selection. But if plasticity has large enough effects, it may suffice to keep the populations persisting despite changes in at least the aspects of the environment that induce plasticity. Now, we know that mammalian trophic morphology is plastic. Orthodontics often relies on this, adjusting the jaw to the forces applied to the bone via the teeth. What we don't know is whether dietary consistency has a large impact on jaw morphology. Now, foods eaten by rodents can differ greatly in material properties, such as hardness, toughness, stiffness. And these properties should affect jaw mechanics as well as the forces produced by closing the jaw. So we know it varies from year to year. In 1950, conifers of the northern Sierras failed to set seeds, and coincidentally, the spring-blooming shrubs lost flowers and didn't produce fruit. So analysis of gut contents revealed rodents ate, and since I can't pronounce that word reliably, I'm just going to call them truffles, insects, shells, and desiccated meat of acorns. So, dietary plasticity in science. Coconut, obviously you know what that is. And not so cute pictures. Yeah, I thought that students send us all sorts of pictures. So empirical estimates of evolutionary rates concluded that on average, about 0.035 sigma per generation in studies that look at 80 generations or fewer. It looks slower if you look over a longer time for reasons we'll actually talk about in a moment. And rates are relatively high in populations exposed to environmental disturbance, but only in looking at phenotypes in natural populations, not if the studies use quantitative genetic estimates or common garden experiments, selecting that the higher rates in disturbed populations are probably due to plasticity. One complication, most studies analyze size measurements, often many of them, and these results can be difficult to interpret. Size can only increase or decrease. Size might increase over 1 or 2 generations, and then decrease, then increase, and then decrease. So in Darwin's finches, that is what happened, and it can erase evidence of prior evolutionary change. So we may be underestimating how rapidly morphologies can evolve when we measure only size. Another complication is the study by Pergams. All these size measurements cannot possibly all be independent. One thing, they're all correlated with size. The rate of change for one trait may depend on the intensity of selection on another, because the rate at which any one feature evolves depends not only on the direct effect of selection on it, but the indirect effect of selection on all other correlated traits. And because they're all correlated, selection may be severely constrained by the structure of covariation. Due to correlations, the population is confined to evolve in a subspace with fewer dimensions than the number of traits under selection. So if we think about this sphere as 3 traits under selection, due to correlations, selection may see only a 2-dimensional plane. So one expectation is that patterns of genetic correlations result in genetic lines of least resistance that can have major effects on long-term evolutionary trajectories. And some studies have shown that for at least 3 million years, populations evolve consistently in the same direction, along the direction of the lines of least genetic or phenotypic resistance. Functional correlations present a very different kind of problem. Functional correlations complicate adaptation because these are the biological interdependencies among parts of a complex. So the optimal phenotype for one trait depends on the phenotypes of other traits. It's no advantage in having jaw muscles so powerful that they break the jaw bones, a robust jaw with teeth positioned so far back that chewing dislocates the jaw, a deep and broad angular process, but a gracile horizontal ramus, and I could go on and on and on. So what makes morphology optimal involves interactions, proportions among all these different traits. So for complex morphologies, correlations may actually facilitate adaptation by allowing adaptively related traits to change in concert, while restricting variation in traits under stabilizing selection. But we know surprisingly little about the structure of variation in complex traits. We expect interdependent traits to covary. But how are those dimensions organized anatomically? Is each part of the morphology a quasi-autonomous module? And you probably all are aware that modularity is now a very hot topic. And are the covariances restricted but broadly distributed? Now, the dominant model among quantitative geneticists for mandibular morphology divides the jaw into the front and the back. If anybody is a functional morphologist, you will understand this is a quantitative geneticist's idea of the two functional modules of the jaw. The two primary functions the jaw serves, according to this theory, is to bear the teeth and to bear the muscles. If this is the case, we might have only two dimensions of variation. Now, this model has very odd implications. First of all, it means resistance to occlusal forces is independent of the forces the muscles generate that close the jaw that bring the teeth into occlusion. So if you think that chewing is a function of the jaw, this separates two parts of a chewing apparatus into two modules. Also means gape and mechanical advantage are correlated. So you can't modify the position or the height of the coronoid process independently of altering the angular process. So we know surprisingly little about plasticity or evolution of complex traits. We want answers to, does plasticity have a large effect by integrating changes across morphology? Can plasticity fine-tune responses to replicate environmental conditions that impose different functional demands? Or is plasticity a more stereotypical kind of response? Is the evolution of complex traits predictable from the structure of covariation? Do complex traits change more rapidly or slowly than simple traits? So the outline for today. First, I'll talk about plasticity, adaptations to dietary consistency using a deer mouse as a model system, because nobody has done this experiment on any Sciurid. Then I'll talk about variation and modularity of Sciurid mandible, how variation is organized. And then I want to look at rates and directions of phenotypic change. Do range shifts, costs of selection, and or intrinsic constraints predict either rates or directions of phenotypic change? And do complex traits evolve at a rate comparable to simple traits? So the data for the analysis of plasticity come from an experiment that Phil Myers ran several years ago. Litters of pregnant wild-caught females were randomly allocated to one of three diets before they were born. This is so the mother was actually fed the experimental diet, so the babies, when they start practice chewing, would begin feeding on the diet they were going to be treated to. So the hard diet was laboratory pellets. There were two soft diets. One was gruel, which was the same laboratory chow that was ground up and mixed with water. The other was powder, which was laboratory chow powdered. So they're both soft, but they're mechanically quite different. The data for the analysis of historical change comes from the Grinnell Resurvey project. And alpinus, speciosus, as everybody knows, have been contrasted for both responses, the range shift responses, and consequences for genetic diversity. Also including Calosporomophilus lateralis, not only because I really like it, because we found that we had a sample that we had measured for a different purpose, And it's much more heterogeneous than the other two, and there are only 15 modern ones. So anybody who is going out into the field for any purpose, make sure you collect the animals. Don't just leave them behind when you're done. We need all of these animals. We have a lot more historical for most species. What we don't have is good samples of modern, and we need them brought in and really well prepared, because when you see what we're doing with them, you'll see why we need perfectly preserved material. So, as you know the story of Tamias alpinus, these are the data that Jim kindly gave us to show different localities and how their ranges have changed over time. So we see is that alpinus has gone through a massive constriction. Speciosus, much more stable in the ranges. And I'm going to call it callow because its common name is way too long as well as its scientific name. And this is from the Grinnell Resurvey report. So, as Emily and colleagues showed, that shifting ranges has led to a loss of genetic diversity. For alpine chipmunk, 6 or 7 microsatellite loci have lost at least one allele, and genetic subdivision is increased. And for lodgepole chipmunk, range stability, there's been no change in overall genetic diversity, allelic richness, or population structure. This is really important for assessing what's happening with phenotypes. Partly because we might assume that a range restriction is going to reduce the population, structure it, have severe demographic consequences, which could be important for predicting evolutionary rates. But this information, Jim sent the paper, which was published the day before I gave my first talk on this material, so can actually now talk about why we might expect changes or differences between alpinus and speciosus in evolutionary rate. So what we're doing is 4 photographs of each individual were taken. 2 on the left, 2 on the right. On each one we measured 15 discrete anatomical points plus a scale bar. And we traced the curves between the landmarks to provide information about curvature of the jaw. And then we averaged the measurements from the 4 photographs. We used individuals that were damaged on one side by just measuring 1 photograph twice. But this introduces both an inflates measurement error and allows the possibility of both random asymmetry and directional asymmetry. So the more of these damaged individuals, in a sense one-sided individuals we've got, the more this inflates the possibility that what we've got is random error isn't random biological error, but measurement error. So our measurement schemes, well, we superimposed all these data by standard Procrustes superimposition, and if you take the course this year or in the past, you will know what that is. We also removed the variation in the semi-landmarks that is not due to biological variation. And after superimposition, we removed half the semi-landmarks we had taken. So what we're doing here is we're removing the variation that's unrelated to shape. It's variation in geometric scale, variation in the position of the photograph under the camera, and variation in its orientation within the plane. So our data consists of 15 landmarks and 85 semi-landmarks. Shape space is therefore 111 dimensional. We don't have 111 specimens of any species, so the occupied space must be less than 111 dimensions. So for the statistical analyses, shape is the dependent variable. And analyses were done using 2 approaches whenever possible. First is to use a permutational MANOVA, in which we calculate the morphological distance between all pairs of specimens, and using the conventional distance metric for shape. And then we apply the model to the interspecimen distance matrix. This is a technique commonly used in ecological studies, which allows you to apply standard statistical general linear models to matrices of ecological distances. This is well suited to high dimensional data, but it doesn't take the variance-covariance matrix into account when it tests for statistical significance. The alternative is a multivariate analysis of variance, which reduces the data, in order to be able to do this, reduce the data to 25 principal components, and then use that set of 25 principal component scores as the dependent variable in the MANOVA. This method does take covariance structure into account. And the multivariate approximations to the F ratio are unreliable when sample size is small relative to the number of variables, which requires restricting the number of PCs to only 25, because that using more would require having more than 100 specimens. Now, I haven't yet found a way to do a multivariate analysis of a nested MANOVA in R or in any other program. So when I do a nested design, the permutational ANOVA is the only possibility. So for study of plasticity, how large an effect can plasticity have? Is the effect large relative to maximal sustainable rates of evolution? And do replicate treatments, gruel and powder, produce the same morphological effects? This is the statistical model. We included both size and sex, as well as diet. And because all members of the same litter were fed the same food, we had to take variation among litters into account in the statistical analysis. This is because these were not domesticated deer mice, and you don't try to cross-foster babies of undomesticated animals. So what I have in bold are the effects that are significant. And diet clearly is significant, and the picture shows the change from the control group mean to the liquid-fed mean. And it's exaggerated tenfold so that you can see it clearly. So to quantify the magnitude of change, you're using Procrustes distance, which is standard distance between shapes, and the Mahalanobis distance. This is equivalent, multivariate version of saying how many standard deviations separate a sample. So it's measured in units of standard deviation. This does take the covariance matrix into account, but it's sensitive to the number of variables. So the magnitude of the effect, control to liquid, Procrustes distance, well, you won't know what this means, but that's a fairly large value. 0.02 is common between species. Mahalanobis distance, well, it sounds large, 4.74, but if I use all the data, it's actually 7.2. That means a change of 7 standard deviations due to being reared on liquid. And control to powder, the Procrustes distance is 0.013. And the Mahalanobis distance, using 25 PCs, is 3.64. But it rises to 6.06 if I use all the data. So the effect of plasticity is very large relative to maximum sustainable evolutionary rates. 4.5 or up to 7.2 versus 0.01 for a generation. It's also very large relative to the average rate of evolution. 4.5 or even up to 7.2 compared to 0.035. Now, the very large effect is probably due to the extreme contrast between the treatments. I mean, feeding them laboratory blocks, which aren't all that hard, but in between sort of seed coat and seed inside and hardness. But then the contrast between acorn and truffles, which a chipmunk may experience in its lifetime, is also very large. Now, both soft diets affect coronoid process, the depth of the angular process, and the posterior molar row. This is actually hard to see. That's an arrow showing the decrease in space between the base of the coronoid and the second molar. But the effects obviously don't look identical. So we want to know is, are the differences in effect larger than we would expect by chance? So we can compare the responses by measuring the difference in response vectors. If two vectors point in the same direction, the angle between them is 0. So we can compare the observed angle between these responses to one we could get by resampling from a single treatment effect. If the observed angle exceeds 95% of the within treatment angles, then we can conclude that the difference between the treatments is larger than we would get by chance. So the observed difference is 32.9. And you can see that the within treatment groups, the differences, the range of angles is smaller. So the responses of the two soft diet treatments differ significantly. One thing worth noting, this is the smallest difference between angles you will see through the rest of the talk. So I think of these as small. So in summary, plasticity can have a very large impact on morphology, equivalent to centuries of evolution at the maximal sustainable rates, or over a century at the average evolutionary rate. And plastic responses vary across replicate treatments, suggesting that the responses are fine-tuned to the environmental factors. So what I now want to look at is the structure of variation and modularity of the Sciurid mandible. How is variation organized? So I want to look at dimensionality, modularity, anatomical distribution of the covariances. So you're going to use principal components analysis, which rotates the data to align it with the major axis of variation. Principal component 1 is the less poetic term for the line of least genetic resistance. It's the major axis of the variation in the sample. PC2 is the next longest axis that's statistically independent of the first. As you will see, they're not at all anatomically independent, they're just statistically independent. PC3 is the next longest, etc. So what we would see if variation is low dimensional is that the first PC explains vastly more of the variation than anything else. We see one enormous value here, and then it rapidly falls off. If you're used to working with morphological data, you might find 2 large PCs, 1 or 2 large ones, everything else is small. If variation is concentrated, nearly all the variation will be captured by a few components, but not by one. If variation is random, all PCs will be equal in length. So this is what we find in the historical population of alpinists. Variation is clearly not one-dimensional. That's 20% of the total variation, as explained by PC1. Variation is also not random. Obviously, way out here, those values are smaller than that. Most of the variation is concentrated in 5 dimensions. And that's true if I combine historical and modern sample together. But it can take 25 PCs to span 95% of the variation in just 60 specimens. So on average, we've got less than 3 chipmunks per PC. That's actually relevant for how selection can act on dimensions of variation. So the anatomy of PCs. Well, the coronoid and angular processes, for those of you who don't know vertebrate anatomy, here we have the coronoid and the angular. That's the incisor alveolus, it sheathes the incisor. There's the molar row. There's the jaw joint. So the coronoid and angular processes and the incisor alveolus covary on multiple PCs in different combinations. Now that means also that how regions covary can't be visualized by looking at any one PC, because they'll covary in different ways on different dimensions. And actually what we have here is a plane, although really it's a 5 space, of lesser resistance. The reason for showing all these shapes is because the PCs, while statistically independent, aren't anatomically independent. The same variables load on multiple ones. So what you see if you look at high scores on 2 and low scores on 1 looks very different. You can't interpret one PC at a time when they have nearly equal magnitudes of effect and they anatomically overlap. PCs have to be interpreted in light of the full space, not dimension by dimension. So in all 3 species, PC1 explains about 20% of the variation. Variation is concentrated in the first 5, but could choose a different cutoff. 25 PCs encompass 95% of the variation, except when sample sizes are very small, because in total you can't have more PCs than specimens. My advisor, who was a student here, once referred to me as being 2 standard deviations out on every axis. If every individual was way out there on their own axis, and so if there were 2 ends of one axis, it would be half the number of axes as there are people. If everyone's out on their own axis, it can't be any more than who's Jim's student. And Bill also. So in all 3 species, we see a complex pattern of variation. And dimensions of the same anatomical regions vary with diverse combinations of others, frequently with the same parts of the coronoid with parts of the angular, differently localized on different PCs. So, is the mandible modular? Well, we can estimate the covariance between the two regions by a Scoffiers coefficient, which is called an RV. Now, this is analogous to a squared correlation coefficient. So when reading the numbers, think about a squared coefficient, not the correlation. We can then test the hypothesis that the observed RV is no higher than we could get from random permutations of the data. So for these three species, the RV between the front and the back of the mandible is always above 0.4. And again, thinking about what that means for squared correlation, not surprisingly, these are highly significant. So this is a case of having two slides that are supposed to say the same thing. So we're now going to look at the front and the back and see how they covariate, the axis that explains maximal covariance. It's like looking at a PC for maximal variance. This is partially squares. And the dominant axis explains 71.5% of the total covariance between the front and the back. And you can see this spread out throughout the jaw. Depth of the angular process, curvature of the coronoid is covarying with depth and curvature of the incisor alveolus. Anyone who thinks about the jaw as serving mechanical function rather than bearing teeth and bearing muscles, this makes sense. So again, in the other two species, we're seeing the same thing. Covariance between angular and coronoid processes and incisor alveolus. So to summarize for variation, variation is concentrated, but not along a line or in a plane. Most of the variation lies in a 5-dimensional space. By most, I mean it's usually at least get up to 50% within 5 principal components. Variation, with larger sample sizes, we might find variation in more dimensions. Variation has a complex spatial architecture. Dimensions of the same anatomical region covary with diverse combinations of other regions. And the spatial structure of covariance argues against the idea that the mandible is highly modular. What we see are covariances of dimensions of variation spread out across the jaw, not quasi-autonomous blocks of anatomy. So now what I want to do is, having set that context, look at rates and directions of phenotypic change. Look at whether do range shifts, costs of selection, or intrinsic constraints predict either rates or directions of phenotypic change. And do complex traits evolve at a rate comparable to simple traits? And again, this was a result of not carefully editing. So our model for the statistical model is shape is equal to size plus x plus time. Time is the factor that we're interested in, which is the contrast between historical and modern. Sex is included in the model because chipmunks are so often sexually dimorphic in size, so they might also be in shape. In this case, the analysis was done both by permutational ANOVA and by a conventional multivariate ANOVA. So these are the results for alpinus. What we see, the bold means it's statistically significant. The brown means it's a term that involves time. So what we see is that everything is statistically significant, and we have an interaction effect. Elevation and time interact. So the effect of time is not the same across low and high elevation, or the effect of elevation influences response to time. All of these factors are significant using a permutational ANOVA. And what we can see is that the differences are in position of the base of the coronoid relative to the molar, the anterior posterior position of the masseter fossa, curiously mental foramen, angular process a little bit here on the condyle. For speciosus, size but not time is significant using a permutational ANOVA, but size and time are both significant using multivariate analysis of variance. I haven't yet figured out exactly why this is true, because it could either be that by restricting the analysis to only 25 principal components, I'm deflating the residual and therefore elevating an F ratio. It's also possible that much of the variation is along lines that don't vary, much of the difference is along lines that don't vary within, and so it comes out as being significant if you look at the amount of change relative to the within sample variation. But one thing that's interesting about speciosus versus alpinus is that the effects are much more broadly distributed across the jaw. For callow, time is the only factor that's significant using both statistical approaches. And again, we're seeing effects that are distributed across the jaw. There's again the base of the coronoid and the molar. Angular process, but we see a lot of change in the condyle. And we see that most of the angular process is affected, as well as the incisor alveolus. So magnitudes of change. Here, comparing the two different metrics, the shape difference and the shape difference scaled by sample covariance matrix. We see is actually alpinus and speciosus undergo the same amount of change. It's spatially very different distributed. It's localized in alpinus. It's broadly distributed across the jaw in speciosus. So everywhere you look, the effect would look smaller because it's summed up over a lot of the landmarks. But the net difference based on Bruchowski's distance is same, and I did a statistical test. That difference is not significant. Lateralis is more, but that may be because our sample is very heterogeneous. We don't have historical from 1 or 2 years, modern from 1 or 2 years. That's actually what we're going to be starting this afternoon, is to get a much more controlled sample of lateralis. But you can see that plasticity does a lot more than 90 years of phenotypic change, or however long it is. This is what happens with size. Size does not do very much. That's size in units of standard deviation. And it's not really consistent from one to another. Only in alpinus is it close to plasticity, but even plasticity doesn't tend to produce that large an effect on size. So, our expectation was maximum sustainable rates for populations in a continually changing environment, subject to fluctuations in the optimum, would be 0.01 per generation. Here we have a problem. We don't know how long the change has been going on. Could have been going on for 90 generations, these breed once a year, or could have been going on for just 35 generations if it's started with warming. So we're going to look at what the expectations are for both times. So 0.9 or 0.35. And as we can see, obviously there's a lot more change that occurred in shape than predicted by the maximum sustainable evolutionary rates. But now using expectations from empirical study of rates that have been estimated over 80 generations and which are therefore comparable to our time scale possibly, is 0.035 sigma per generation. So over 90 generations, that's the amount of change you might expect. Over 35 generations, which does strike me as more reasonable for these, since studies that have looked at what happened between, let's say, 1950 and 1970 didn't find changes. But you can choose whichever value you prefer to look at. And we find is that shape, if that is the reasonable estimate, is exactly where we would expect it to be. Size is smaller amount of change than we would expect.
That's a question. The 0.01 standard deviation is per generation. So it's kind of relevant to interpreting all of this. So what is that an expectation for some probability of stochastic population extinction over some number of years? And what is the actual time scale to which that's supposed to apply?
Well, what they did was they took earlier models by Lynch and Landy and dealt with the possibility of stochastic demographic shifts. So if you conclude stochasticity in demographic shifts and then look at synergistic effects of carrying capacity and rate at which the environment changes, the magnitude of the fluctuations from year to year in the optimal morphology, what you find is that many of the effects are multiplicative. So if you have populations that are experiencing a continual shift in the optimum and undergo fluctuations from year to year, and there's always the probability of extinction just due to random stochastic effects, then that would be the maximal sustainable evolutionary rate that they would expect. It's about 1/10 of earlier estimates. But since the earlier estimates didn't take either stochasticity of the demographics, which is obviously important when populations are shrinking, and it didn't take the combination of a moving optimum and fluctuations around the moving optimum, which seems to be the earlier models, which were more optimistic, were also pretty unrealistic. Does that answer the question?
Yeah.
So, summary on rates of historical change. Both mandibular size and shape change over the past 90 years, by far less than could be accomplished by plasticity within a single generation, by more than expected from the theoretical maximal sustainable rates, by less than expected for 90 generations at the average rate, but by just about what's expected for 30 generations, 35 generations at the average rate for a disturbed population. So the amount of change does not appear to be predictable from range shifts. Alpinus and speciosus undergo statistically indistinguishable amounts of change, and both change less than callow. But as say, callow is much more heterogeneous sample. So, I mean, based on at least our estimate from what we have now. Shape changes far more than size for these. Perhaps due to more heritable variation in shape, more intense selection, or what seems most likely, fewer reversals. So predicting direction of change. We're now going to compare the direction of change to the lines of least resistance. And since we don't have a line of least resistance, we'll look at directions of change relative to PC1 through PC4. And we'll look at whether the changes in different species, whether the change in one species predicts the change in any other. And again, these comparisons are done by measuring the angle between vectors and testing them for statistical significance. So here's for each species against PC1, PC2, PC3, PC4. Maximum possible is 90. Since the orientation of a PC is random, if I got a value that was higher than 90, just flip the PC. So you can see there's just about perpendicular to every axis of variation. Phenotypic change is clearly not aligned with any line, plane, force space of greatest variation. Now this is what we actually would expect from adaptive evolution in a high-dimensional space. Under genetic drift, the expectation is that drift will occur in the direction of greatest variance. Constrained selection, expectation is change will occur in the direction of greatest variance. Clearly, that is not happening. So to look at the effects on of the three species, between the species, these vectors could go to 180 degrees if one was changing in one direction and the other was undergoing the absolute opposite changes. For alpinus versus speciosus, 72. Alpinus to lateralis, it's 93 or 94 degrees. Between speciosus and lateralis, it's 102 degrees. Sort of obviously, these species are not changing in the same way. So for summary for directions of change is not predictable from the lines of least resistance or the changes in other species. I'm trying to figure out if it could be predicted by altitudinal gradients, but the signal from elevation is significant only in a case of alpinus, and it's too slight to actually be detectable statistically as a direction of change. So what may be predictable is the unpredictability of phenotypic change. Changes in one species are likely to parallel the changes in another if what these changes reflect are local biotic interactions and not the driving force of a single climatic variable. So what we would expect is the same species in two different localities, unless there's extensive gene flow between them, would be undergoing changes that are just as different as we're getting between these species. Hence the desire to do callow from different localities where we have large enough samples, because somebody collected them, hint. So conclusions. Rates of phenotypic change for shape are far lower than what could be accomplished by plasticity within a single generation. But they're high relative to the maximal sustainable rate and on the order expected for populations in disturbed environments. Plasticity may enable species to adapt to fluctuating environments without incurring the demographic cost of selection. So the reason why the average rates estimated on phenotypes and ours estimated on phenotypes, may greatly exceed maximal theoretical rates, is that plasticity is not necessarily producing the morphology we're seeing, but it's enabling the populations to persist despite these rapid environmental fluctuations as they respond to the more sustained climatic change. Rates of phenotypic change for shape are close to the average evolutionary rate for species, but size exhibits far less change than expected from empirical studies of rates. As I say, size may have less heritable variation, as seems unlikely, be under less intense selection, which seems even more unlikely considering how important size is physiologically, also in terms of biotic interactions, or it undergoes more reversals than shape, which I think is the reason why we can see such small values for size instead of shape. So, conclusions, the directions of change are less predictable than rates. Evolutionary rates may be constrained by the demographic costs of selection, whereas directions depend on the directions of selection. And that variational complexity may enable multiple combinations of traits to be selectable, and thus make non-modular functional complexes evolvable. So where do we go next? Well, into the collection. To photograph more Sciurids, see if a broader analysis will find that rates are more varied and therefore not predictable, or if they are predicted by range shifts, just not in this case, by elevation or both, and to see if directions might be more predictable. And then back to Michigan to measure the nearly annual samples collected of Peromyscus in the late 80s, early 90s, to get better estimates of per generation rates. And Leucopus and Miniculatus would show contrasting range expansions and contractions. So, acknowledgements for the plasticity experiment, Phil Myers. Curators, obviously, both Phil Myers and Eileen. Collections and advice, Jim Patton, Chris Conroy, and Emily. Europe undergraduate students who did enormous amounts of work. Access to unpublished data, encouragement, etc. And I put etc. because the list would get far too long for a slide, Jim. And support from the Museum of Vertebrate Zoology. Thank you.
Put the lights on, please, and then we'll take some questions. Someone push the lights, please. Okay, so a question. No.
In cases like speciosis, where the change is kind of more general, are you able to interpret this in any way in terms of what it's responding to? Because
I
it's
would
not
love
out
to.
there.
The only thing I could compare it to at this point was to look at the vectors of changes for plasticity in Peromyscus, because that's probably generalizable since it's showing, in a sense, what we expect from studies of humans and from dogs and pigs, where the changes are localized can be predicted essentially by biomechanics. The differences between these Sciurids and Peromyscus diet vectors are smaller than the differences between the Sciurid vectors, and a lot smaller than between PCs, but they're not small enough to say a dietary consistency. So at this point, given all that we don't know about ecology, especially of alpinus and speciosis, I really hesitate to interpret this. I mean, I'm hoping that between the analyses of the isotopes and the behavioral observations being done now, that we'll know enough about at least what's going on now to put together with Grinnell's field notes and see if we can figure out what in the environment might have changed that might have driven these changes. Obviously, we would love the answer to that. Yes?
If you had, so we live in a real world, unfortunately, but if you had, the virtual world, what kind of sampling, well, I mean, not size, obviously, the more the better, but what kind of temporal sampling would be optimal to be able to address,
You'd hate to know the answer.
No, I don't. I wouldn't hate to know the answer. The Cowfish and Game may hate to know the answer.
I think
I'll
at
go out and
least
get 'em.
for 5 years, we need annual.
Annual?
Because what we need to know is if they flip back and forth every year, if, at least for some populations, they're continued over 5 years, Then if we're seeing continued over 5 years, then we could start saying, look at 10. But at this point, we don't know if they're flip-flopping. I mean, we saw with those Darwin's finches, you can go
Yeah.
flip, flop. And the grants looked at 30 years and said how unpredictable things were, and that essentially is erasing. But then they said, no, it's not actually erasing it. The peaks may erase depth and shallowness, but pointiness persists. Plus, one real difference with Darwin's finches and anything I've ever dealt with in my life, heritabilities for those beak traits are like 0.7, 0.8. So every little tiny bit of selection produces evolutionary change. We're not going to see anything like that. So I think we can't just see it in the phenotype. We're going to need to look generation by generation. Yeah.
In that vein, in the Permiscus data, do you have any information on the fitness correlates of these traits that you're measuring?
Well,
Can
in the
you use
case of
selection?
Permiscus, since it was lab populations, and they were all from a single population from Ann Arbor, and then they were just bred. Well, pregnant females were brought in, their offspring were bred, and then it was those offspring that were fed diets. Now, this may be gut instinct rather than purely rational argument, but I'm sure I could produce a rational argument. Plasticity is producing the optimal morphology for a given environment. So natural selection and plasticity ought to have exactly the same effects, if selection is being effective. Since plasticity is adapting the morphology to the exact environment in which it finds itself. So that's
But that argument presupposes, so plasticity is a trait that can evolve in response to stabilizing and disruptive selection. And so that presupposes that plasticity is perfectly, that plasticity is at the adaptive peak, its adaptive peak.
Well, let's talk about
Which-
the more specific case, because the question of whether plasticity is a trait, I mean, I've read enough of the Via and Pigliucci and Shiner papers to realize that when they're talking at the level of theory, I have no idea what they're talking about. So we can talk about jaws. And what we know is producing the phenotypic response is that bone cells can sense the mechanical forces applied to them through, as far as we know, the little caniculae. So the cells perceive distortion, deformation.
And that is a trait.
In response, I mean, there's actually good evidence in a second. In response to that, bone is deposited, or if it's unloaded, it's resorbed. So we sort of basically know the mechanism. There's a fascinating discovery, which is that one of the standard wild-type populations of mice doesn't respond to either loading or unloading. And so they've done QTL studies to look at the differences between these different populations of mice. 390 genes are expressed in response to this bone deformation in, I think it's Black 6 and CD1. Black 6 we all know is bizarre. But in the C3H mice, that's the difference and that's based on the last microarray data that I know of. So there certainly is a genetic basis for variation in plasticity. And whatever that is mediating the expression of at least 390 genes that are involved in producing bone or resorbing bone.
Which is why you have to use quantitative genetics to study it.
As opposed to, you mean pop
Single
gen,
gene.
single gene? Yeah,
Yeah,
yeah.
yeah. But it's, you can still think of it as a trait. I mean, that whole argument between VA
Yeah.
and blah, blah, blah.
Had to
That
do
was a
with
statistical,
polynomials.
yeah, exactly. That was a statistical artifact.
I mean,
The reality is that any trait you decide to call a trait, you can treat as a trait. And, and so plasticity is an extremely complex trait. It's just it's complex in a different dimension than all these other dimensions that you're adding dimensions, which is the environmental dimension.
Well, I think it's using the same dimensions that selection would use, because a selection could produce the optimal morphology for a given set of forces on the jaw. Then it would be producing bone resorption and deposition where mechanical forces are applied. Well, that's what plasticities do.
Right, and so it may be that plasticity is an extremely conserved trait that
Yes.
isn't going to evolve really rapidly in response to climate change. But on the other hand, it might be that plasticity will, I mean, that gets to this question of the sort of
G times E?
the rapid changes in directionality of selection from one generation to the next.
See, I don't,
If you have a lot of that rapid change, then plasticity is going to be important. If you don't, it's not.
we don't know how much genetic variation there is in plasticity in natural populations. I mean,
Right. Yeah, and that's, you know, when you talk about things that are really hard to get at.
But, you know, if we're
That's
seeing
one of them.
7 standard deviations of difference between pellets and liquid, I don't know if they have to be any more plastic than these Peromyscus are. Because, I mean, to put it technically, that's a whoppingly large change.
Yeah.
Okay, I'm going to draw to close this fascinating discussion. Plasticity is really important and huge, as you've shown here.
And
So-
really hard to study.
Yeah.
Really hard to study, exactly. But fun to argue about. So, thank you very much, both of you.